This is lecture 30, anabolic signaling, part 3 of 7. And we're going to begin where we left off with the last lecture, some diagrams where it shows both the big picture and the small picture. So we see a lot of interactions, but they should be starting to make some sense. You should start to recognize a lot of what's going on here. Not everything, not every enzyme, but more and more of it should make sense as we get further and further into this lecture series. And here's another, you've seen it before. It's a good one though, and we're going to keep working our way very gradually through a lot of this stuff. And the last one, before we get started on today's lecture, this is the whiteboard practice session from memory of some students, no sources. So a few students a couple of years ago, this was their practice session. And it's a really good diagram. So some of the stuff that you don't recognize here, we're going to cover today. Okay, we'll move forward. In the last lecture, we covered mTOR pretty thoroughly and discussed its purpose, right? The preservation of fitness, of metabolic fitness, how fit, right? How appropriate or compatible is your metabolism to the environment in which is doing its metabolizing. So, what are the conditions outside of my cell walls? mTOR wonders. How much food is available? How much muscle is it going to take to acquire that food? And so once mTOR figures this out by vast signaling networks, it matches the cell's biomass to its functional need as precisely as it can. So that's what I mean by fitness. So in this lecture, we're going to talk about some of the inputs for mTOR. We'll talk about the upstream phenomena that regulate mTOR activity. And in a future lecture, in a couple of future lectures, we'll do applications, but this one is activations. And those activations, these are diverse. mTOR's seemingly disorganized organization allows it to respond to a variety of upstream inputs, not just like did you eat protein, right? But other nutritional inputs, hormones and growth factors, mechanical inputs, other forms of stress. We're going to start going through all of that stuff. But we'll split it into 4 categories, these 4 categories, and we'll go through them in order. So the first one up is the immune system and other chemicals, inflammatory lipids and proteins and that stuff. And then we'll do mechanical loading, how the tension applied to tissue transmits its own cascades independent of hormonal and chemical receptors. And then the next lecture, we'll do those 2 today. And then the next lecture, we'll do the endocrine system, not just stuff with like hormone-like effects, but the actual hormones themselves. And then we'll also do nutrition. So you remember arachidonic acid, polyunsaturated fat, maybe it accounts for 15%, give or take, of the fatty acids in your muscle's phospholipid bilayer. And if you exercise, you'll release some of it. You can also take arachidonic acid as a supplement, and you will be punished with soreness, but we'll hold off on that until we're talking about applications. So arachidonic acid, this gets converted to prostaglandins through cyclooxygenase or COX. In the last block, we talked about nociceptors, pain fibers, nociceptors, and the role of prostaglandins in signaling delayed onset muscle soreness. There are other things involved too, but prostaglandins are the major contributor. So now let's talk about the role of prostaglandins in hypertrophy. Um, when prostaglandins bind to their cell surface receptors, they signal MAPK, mitogen activated protein kinase. And this pathway runs parallel to PI3K, PKB, mTOR. Um, and they eventually, the MAPK pathway, it eventually arrives at that very anabolic RSK that we covered in some detail earlier. Now, if you take NSAIDs, non-steroidal anti-inflammatory drugs, NSAIDs, they inhibit cyclooxygenase. And aspirin is a suicide inhibitor of that enzyme, an irreversible COX blocker. So you reduce soreness, but you also impair tissue remodeling. You reduce that at the same time. This is the guy who discovered how aspirin suppresses prostaglandin production. He's a night bachelor, which is the lowest ranking of night. So like Elton John and Paul McCartney, those are real knights in case you want to go to war and you're looking for, whom to bring. Uh, so here's a closer look at prostaglandin signaling, again, running parallel with mTOR signaling. Uh, Prostaglandin production and prostaglandin signaling, MAPK signaling, that's all in green. mTOR is uncolored, mTOR is in white. And you should recognize pretty much everything here. PLA2, arachidonic acid, PLA2 is phospholipase A2, arachidonic acid, COX, prostaglandins, and then notice the crosstalk with mTOR. You're going to start growing if you can also activate mTOR. So focusing on the signaling itself, ignore the production of prostaglandins, but look at the signaling itself. When prostaglandins bind, we see MAPK or ERK signaling, mitogen activated protein kinase, or extracellular signal regulated kinase, MAPK or ERK. Again, this is just another phosphorylation cascade, different from the PI3K, PKB, mTOR cascade. But again, it runs parallel and it interacts. When you get to RSK, P90 ribosomal S6 kinase, you have some downstream convergence between the two cascades, but you also have intermediate interaction between them. So you see tuberin being inhibited there. And another interaction that isn't here, that is not shown here, but exists in real life is Raptor. MAPK signaling will help activate Raptor. So tuberin gets inhibited, Raptor gets activated. These are both points of interaction for MAPK to help activate mTOR. And here's another diagram showing the interactions between MAPK and the PI3K, PKB, mTOR cascade. You don't need to know the detail of MAPK, but to be a completionist, RAS, RAS is a GTPase, RAF, RAF is a kinase that activates MEK, mitogen activated kinase, and then MEK activates ERK and or MAPK, and then it goes all over the place, right, including RSK. And with that, pretty much everything here. I mean, RAG, which you see connected to mTOR, that's amino acid signaling, and we'll talk about that in detail later in a future lecture. And then at the top, you see PTEN, PTEN, phosphatase and tensin homolog. This is the opposite of PI3K. It converts PIP3 back to PIP2. Other than that, you know everything here. And this is a good article that talks about some of the sites of interactions. MAPK signaling suppresses tuberin and promotes Raptor. When you see SER 863, that's a phosphorylation site. If you see serine or threonine or something in a number, that's just a particular location of phosphorylation. You don't need to know any specific locations, just that it gets phosphorylated and whether that phosphorylation activates or deactivates. Now, if you go to the gym and beat up a bunch of tissue, you'll get a big chemical response, a big inflammatory response. And it's not just prostaglandins. Among the chemicals that get released is IL-15, interleukin 15, inter meaning between, leukin as in a leukocyte. So IL-15 is a cytokine, a small signaling protein that's released during damage. And lots of studies have examined the relationship between IL-15 and hypertrophy. So in the presence of IL-15 overexpression, you translate a bunch of myosin and other proteins. And at this point, you should be able to guess the pathway. I mean, you know it's not going to be PKA, right? That's catabolic. PKA is catabolic. And IL-15, we're looking at anabolic pathways here. So it's either MAPK or mTOR, right? Those are your available guesses. In this case, you see a lot of mTOR activity in the presence of IL-15. The more damage you create, the more IL-15 you see. The more IL-15 you get, the more mTOR signaling you have. The more mTOR signaling, the more hypertrophy. So looking at a diagram of the signaling, you see PI3K activation on the left. You also see JAK-STAT getting signaled too. We talked a little bit about that one. In the center there, and then MAPK on the right. But the major signaling cascade for most of this stuff is PI3K, PKB, mTOR. And here we see IL-2, right? Interleukin 2. When this binds to its cell surface receptor, you see PI3K activation. That's the road a lot of cytokines take to exert their metabolic effects. Here we're looking at interferon gamma. It's made by a lot of cells, but largely white blood cells. It's another cytokine. Um, it doesn't show it here, but it also does go through JAK-STAT and MAPK. Other stuff on this diagram that you should recognize, right? Insulin signaling and growth factor signaling. Just think like insulin-like growth factor, fibroblast growth factor, vascular endothelial growth factor, a bunch of factors. Factors are proteins. This is just a bunch of proteins. But we're usually thinking of IGF here, insulin-like growth factor. Then you see TCR, that just means T cell receptor. CD28, that's cluster of differentiation 28, protein expressed on T cells. You see IL-2 there. And then there's a chemokine receptor, cytokines that direct chemotaxis. So most of this stuff you don't need to know. Just pay attention to interferon gamma and IL-2 on this slide. Still on the subject of interferon gamma, the title of this article says enough. Endogenous, right inside of you, interferon gamma is one component among um, like tons of these things that's required for, quote, efficient skeletal muscle regeneration. Um, and so for skeletal muscle remodeling, this is one of a bunch of endogenous chemicals. Myostatin is something we've also talked about before, another endogenous. This is all endogenous stuff. Um, a lot of autocrine action here, but also some paracrine and endocrine signaling with myostatin. And so you should remember myostatin and follistatin. Myostatin is a myokine, right? It's a cytokine, a protein produced by muscle fibers, and it blocks PKB. So it inhibits protein synthesis, and it also inhibits the differentiation of myoblasts, so you don't get mature muscle fibers. And then follistatin, if you remember, is a binding protein, and it binds to myostatin and inhibits it. TNF, tumor necrosis factor, another cytokine, pro-inflammatory. It's released by macrophages, and it activates both PI3K and MAPK, and you can see that signaling in the diagram to the right there. Wnt proteins, we've talked a little bit about this, wingless related integration site, Wnt, right? WNT, but pronounced Wnt. And a lot of Wnt proteins are made in epithelial cells, and these can upregulate or downregulate mTOR. So Wnt binds to a frizzled receptor, passes its signal to a disheveled protein. It's a G protein coupled receptor. And so disheveled, it gets activated. And um, it gets its name disheveled from flies. Flies that have a mutation there have disheveled hair. But for mTOR activation, this is the only part you really need to know. Wnt proteins inhibit glycogen synthase kinase. And that glycogen synthase kinase turns off glycogen synthase and turns on tuberin. So through Wnt proteins, you get some activation of mTOR and some encouragement for glycogen production at the same time. You stop the inhibitor of glycogen synthase. Reactive oxygen species. These are the things that you take antioxidants to get rid of. So you have vitamins C and E. But if you take a bunch of that stuff around your workouts, if you take a bunch of vitamin C and E stuff around your workouts, you can impair mTOR signaling. And lots of studies have evaluated the relationship between reactive oxygen species and mTOR. Um, what are the metabolic effects of increasing or decreasing levels of reactive oxygen species? The relationship with mTOR is kind of complicated. Slightly elevated levels are anabolic. So increase your levels of reactive oxygen species a little bit, and that's an anabolic signal. Hugely elevated levels, though, that's catabolic. So there's a range that you want to be in, a slightly elevated range. And that's it for the immune system and chemical activation, or in some cases, inhibition, like myostatin, of mTOR. And here's a list of the chemicals we talked about, the ones that you should know. Um, for the most part, the signaling of all this is going to go through PI3K, PKB, mTOR, but also you do see MAPK signaling. All right, let's move on to mechanical loads as the stimulators of mTOR. Your mechanical sensors sense all of the characteristics of a load, weight, speed, and range of motion. Everything quantifiable gets quantified. And the three notable mechanoreceptors are titin, cadherins, and then especially integrins. So all of everyone, we're all like good physicists unconsciously. So think about like a center fielder. Um, let's say it's softball. And so she sees the ball leave the bat and runs to exactly where the ball is going to land. If you gave that same person a pen, okay, and a piece of paper and all day to figure out the geometry of the arc and consciously calculate where the ball would land, she probably couldn't do it. But unconsciously, she figures out in like a second. Like a Golden Glove outfielder can unconsciously calculate trajectories of the ball, like before the batter has finished the swing. Your unconscious body is an amazing physicist. And the roles that these mechanosensors are playing are doing just as impressive a task as the center fielder, and like astronomers and whatever. Um, so the mechanisms of how this stuff is works. Notice the integrins here in the center of the diagram. There's an integrin, a labeled integrin there. You have more than just actin and myosin in your muscle. It's not just those two contractile proteins floating there, like magically in your muscle interacting, or you have a bunch of other structural proteins holding everything in place. And those structural proteins aren't just support beams, right? They notice stuff too. Many of them notice stuff too. So these are your internal physicists that are crunching the numbers of all of your movements, your motions, your force output. They collect information about whatever activity you're doing and they relay it. Um, and so this picture should make some sense. The extracellular space, right, with its collagen and fibronectin. Remember, if you're going to form a clot, right, you get that fibrin fibronectin cross linking. So you've seen this stuff before. Then you have a cell membrane, and on the inside of the cell, you have a cytoplasm. You see actin, you see alpha actinin. That's the protein that holds your actin in place. It binds it to the Z disc. But the integrins, these are transmembrane proteins. Transmembrane, meaning it goes through the membrane. There's something on the inside, something on the outside, something in the middle. So integrins are relaying information from the extracellular matrix to the inside of the cell. And this is a picture of cardiac. It's not a picture. It's a figure, a diagram of cardiac muscle. But we're looking at the same stuff. Right? So let me walk you through what's in it. What's in the diagram? The costamere. You see that word that connects the sarcomere to the sarcolemma. A ton of proteins and glycoproteins. Um, and then, of course, you see the sarcomeres themselves. That's what we're looking at. Here's strings of sarcomeres. I mean, we are looking at myocytes, which just means muscle cells. But what to pay attention to here is titin, one of them, which goes from the Z disc to the M band. You see it inside the sarcomere. It's about 30,000 amino acids long, a big protein that serves as the rubber band of the skeletal muscle. And then integrins, we know what these are. They're transmembrane proteins, the red things on the right half of the diagram, that cell. Integrins are noticing the extracellular matrix and relaying that information to the interior of the cell through focal adhesion kinase, FAK. It's labeled focal adhesion complex here, the orange dots, but that's a kinase that initiates phosphorylation cascades. And then there's cadherins, um, calcium adhesion, right? Adhering with calcium, cadherins. These are another transmembrane protein, and they connect cell to cell. They connect to the actin cytoskeleton, right? In part of a it's a part of a protein complex with catena is with catenins, right? What you see here, the catenin, that lavender colored dot. That's how you're connecting cell to cell and going to the actin. So all three of these are mechanosensors, and um, the cadherins are intercellular loads. That's what they're sensing. Cell to cell communication, intercellular loads. Titin is sensing intracellular loads within the cell. Titin is not a transmembrane protein. The other two are, but titin isn't. It's just sensing the inside of the cell, inside the sarcomere, intracellular loads. And then there's integrins, extracellular loads from the extracellular matrix to the inside of the cell. That's what integrins are sensing. And then here's Titan. You've seen this image before in the first block of this class. You saw this exact image. It spans half the sarcomere, and it's not just producing a stretch recoil response. It's not just a rubber band. That's the context we talked about in the first block. But Titan is also noticing stuff and relaying messages. Among its functions is mechanotransduction, cell signaling cascades that are initiated through mechanical stimulus. And again, titin is not a transmembrane protein, but integrins and cadherins both are. If you've ever, there's a little hermit crab on the screen here. If you've ever gone up to one of these, it has its like feelers out there. It's like checking out, it's like surveying the environment, and then you come up and like poke it with a stick or your finger or something, and it sucks its feelers back into its little shell. That's sort of what some of these proteins are doing, right? They're inside of the shell, right? The cell. But they've also got their feelers poking out of the shell, right? The cell. And they're detecting information about the external environment and they're relaying that information inside. It's like, are we safe and can we relax? Or is this stressful? Is this dangerous? Is it hot? Is it cold? What's the environment like? So think about your appetite. That's all your appetite is doing. It's reaching out into the environment. Is there food available? Just check in, right? That's what your appetite is doing. Um, and the integrins do a just check-in with the extracellular matrix, and then cadherins. Again, those are cell-to-cell communication. Cells, super social. Cells are not isolationists. They communicate with each other. And so cell to cell communication is sometimes referred to as cellular crosstalk. So the bridge here would be a cadherin performing that cellular crosstalk. Looking at the mechanisms for mechanical healing here, mTOR, right? So think about Alfredson's, the heel drop protocol. This is activating mTOR in the tendons through integrin signaling. So around 2008, 2009, studies started coming out that were showing mechanical loading a la carte, right? It's, like a acapella act, just all on its own, mechanical loading can do signaling. It can do hypertrophic signaling, right? It doesn't need hormones and chemicals to do it. Now, there's no shortage of literature from the early 2010s and beyond up to today talking about mechanotransduction, not just as a signal for remodeling of tendons, but hypertrophy for skeletal muscle too. And tendons, though, getting back to this idea of tendons and Alfredson's, tendons don't have inflammation. So terms like tendinitis are a misnomer. Itis means inflammation, and where is that in a tendon? Um, and so there's tendinopathies is how we describe it today, but not tendinitis. And so um, tendons aren't getting the same stimulus that skeletal muscle itself does uh, through all the mTOR activating cytokines. And so without inflammatory healing, you have to do something else to remodel and heal a tendon, and that's integrin signaling. So that's what the Alfredson protocol is doing. That's what it's all about. It's about effective integrin activation. It just also happens to cause inflammation in the skeletal muscle. So you will feel soreness in the skeletal muscle, but that's not a signaling that occurs in the tendon. So here we're looking at signaling of through rather, I guess, of the integrins, and they provide an upstream signal for mTOR activation. You see FAK, right? That's focal adhesion kinase. Downstream from that, you see PI3K signaling, and you see MAPK signaling on the far right. And the far left, it's in both. MAPK you see on both, but sort of in the middle there, you see PI3K, PKB, mTOR signaling. And focal adhesion kinase is also a hub for integrin cadherin crosstalk. So there are lots of ways that mechanical loading will induce hypertrophy. We've covered PI3K and MAPK. In this diagram, you see uh, PI3K and ERK, which is just that's MAPK. But one additional thing on here is IGF-1. MGF, mechano growth factor, is a variant of IGF. Mechano meaning mechanical loading. So you load the cells and they release MGF, mechano growth factor, and you get autocrine signaling. So most of your IGF is coming out of the liver, but MGF, this variant, you're releasing from skeletal muscle in the presence of loading. So you'll get some additional PI3K and MAPK signaling that way as well through MGF in response to mechanical loading. More PI3K than MAPK though, just because in the diagram you see, both PI3K and MAPK signal doesn't mean they're signaled evenly. It tends to be more in lots of these, especially like IGF, it's more PI3K. But there's another pathway on the diagram here that you see turning on mTOR through mechanical loading, and that's DGK, diacylglycerol kinase. Sometimes you'll see it as DAGK, but DGK is another way to activate mTOR, and it doesn't go through PI3K or MAPK. What DGK does is phosphorylate. It's a kinase, right? It phosphorylates diacylglycerides. So like a triglyceride minus one fatty acid, a diacylglyceride. You should remember this, where um, adipose triglyceride lipase has lysed one fatty acid. And so you have two remaining fatty acids on the glycerol. That's a diacyl glycerol or a diglyceride. DGK, the kinase, phosphorylates that, converts it into phosphatidic acid, PA, phosphatidic acid. And that stimulates mTOR. And this process is triggered by mechanical stimulation. So here's a 2013 article where researchers were discovering that is DGK synthesizing the PA from diacyl glycerides. And the PA is initiating skeletal muscle hypertrophy. The PA actually binds to mTOR. And so previously, researchers were talking about PLD, and then figured it out, a decade ago. So here you can see the diacylglycerol or diglyceride and DAG kinase or DAGK or DGK. That just puts a phosphate on it. That's what kinases do. They phosphorylate things. So a lot of eccentric loading, and you're going to see an increase in phosphatidic acid, PA, content within muscle cells. And then PA activates mTOR and increases the hypertrophic response to exercise. There are a couple of um, mechanisms of how phosphatidic acid can activate mTOR, including binding to the FKBP12 domain where rapamycin binds. But a question, does the same thing happen with oral phosphatidic acid supplementation? Because you can take it orally, you can take it exogenously. Um, and does that do the exact same thing as endogenous mechanically stimulated? We'll get to that, not today, but when we get to the practical application section, we'll talk all about that. And then one last mechanism that we'll talk about is stretch activated channels. With eccentric loading, you'll get these SACs, stretch activated channels, opened up for some calcium influx, and you see a subsequent augmentation of PKP signaling. Another diagram where you see SACs on the far right, permitting the influx of calcium and facilitating PKB activation. Although in this one, you see that PLD on the mid left side, that should be DGK. So PLD is phospholipase D. We've talked about phospholipase A2, right? We're going to release arachidonic acid. Phospholipase D it does make phosphatidic acid out of phosphatidylcholine, but that's not a pathway that's really induced by mechanical loading. DGK is, diacylglycerol kinase is, so that should be a DGK there. And here's a big picture look at mechanical loading. It triggers a lot of stuff, IGF dependent and IGF independent pathways are all activated. So one question you should be able to answer by now, if you knock out mTOR with rapamycin and you load muscle mechanically, what happens? Do you grow in that situation? No, you don't, right? Even mechanical loading, I mean, just the mightiest, most comprehensive of the inputs for mTOR is sensitive to rapamycin. Um, Wortmannin, though, this is a PI3K inhibitor, phosphatidylinositol 3 kinase, right? It converts PIP2 to PIP3. And Wortmannin inhibits it. So if you take it, mTOR signaling by most pathways, not all, but most pathways, is blocked. I mean, just a massive reduction in mTOR activity. But diacylglycerol kinase, DGK, doesn't go through PI3K. And you see that on the far right. And so most signals for hypertrophy are abolished in the presence of Wortmannin, but you can still sneak some signaling through using mechanical loading. And in pinkish red on the left, you can see AMPK's activity, um, the effect it has on both tuberin and raptor. It inhibits raptor and it promotes tuberin. So AMPK is shutting off mTOR. We're going to cover AMPK in detail in a future lecture, but for now, just know this the primary endogenous inhibitor of mTOR. And you could say that rapamycin is the primary exogenous inhibitor because we don't make rapamycin endogenously. We can take it exogenously to inhibit mTOR, but AMPK is an endogenous inhibitor. And at the very bottom, you see VPS34 being inhibited by wortmannin. So what that means is VPS34 is a type of PI3K because that's what wortmannin is inhibiting. So this is a type of PI3K. Leucine, the amino acid, activates it. You don't need to know about that. But in the presence of wortmannin, most signals for mTOR activation get knocked out. Nutrients is a little bit different. I don't quite like this diagram as is. Those nutrients signal a little bit differently, but we're going to talk about that later. But growth factors, right, including both immune system chemicals and hormones, which we'll talk about in the next lecture, all of that stuff really gets shut down in the presence of wortmannin. But mechanical loading still triggers some growth. Rapamycin, though, that's downstream from PI3K, downstream from PKB. That's knocking out mTOR itself. Mechanical stimulation of hypertrophy gets wiped out by rapamycin. Now, we've been talking about these different upstream activators of mTOR in isolation, like immune chemicals. That was one topic, and mechanical tension. That's a totally different topic. But there is overlap. There are relationships between chemicals and mechanical tension. So integrins, just as one example, integrins seem to trigger reactive oxygen species, which can affect cadherin. So there's chemical mechanical overlap. That's just one example. You don't need to know. I just wanted to illustrate that nothing really exists in isolation. There's always interaction. All right, so let's summarize mechanical tension. We have cadherins. These are intercellular signaling. We have titin, right? This is intracellular signaling. And then we have integrins, which do extracellular signaling. You've seen this picture, but it's a good look at all of these mechanosensors. It's not all of them, but the major ones that we're talking about. And it gives some indication of where they do their sensing. And these are the pathways that mechanical loading works through. PI3K and MAPK through both focal adhesion kinase and through facilitating MGF release, again, mechanogrowth factor, a variant of IGF. Um, and this is autocrine signaling through those same pathways, mostly PI3K, but also MAPK. And then also DGK to make phosphatidic acid and the stretch activated channels to permit calcium influx. And that's it for this lecture. In the next one, we'll look at the endocrine system and nutritional regulation of mTOR. And here are the review questions for this lecture. What you should pay attention to, sort of your keenest attention, should be assigned to these. And I'll see you in the next one, in the next lecture, when we work through the mTOR interactions with hormones and especially with food.